gentlemen, first of all, I want to thank you for your attention. I'm in Oviedo. I come from Georgia. I'm currently attending a school called Georgia Gwinnett College. I'm going for my bachelor's in cell biology, and I'm working under Dr. Sahani this summer. And as you can see from the title of my project, the subject of interest is Hexacyclis aurifolia, also known as wild ginger. Now, this wild ginger is found all over the southeast U.S., and in fact, it's found only in the southeast U.S. There are other types that are found in Europe and Asia, but this specific species is only in this area, indigenous to this area. Traditionally, it's been used as a flavoring in tonics by Native Americans, even went as far as to the belief that it could even cure heart problems. Even if you were to go today and do a quick internet search, you'd find that there are certain recipes and certain things that say you can substitute ginger for this. However, it's a little bit odd because on those very same websites you see this sort of caution about its toxicity. Upon looking at the chemical composition of the root extracts, it gives us a better picture of why. Now, you see I listed the top 4 molecules that is found within the extract of the roots. I want to mainly focus on saffron and beta-asterone, and I'll get a little further in depth in that as the presentation goes on. But for now, we know that saffron has almost 60% of the extract, which leads me to forming my hypothesis. Saffron has been known to be a carcinogen in rats. Tests have shown that it forms tumors in liver cells of test rats. But in a sort of weird way, the same carcinogenic effects have not been linked to humans yet. What has been done on human studies is in vitro or in culture or in glass, as it said. What's been found is that certain cultures, such as human leukemia cells, human cancerous oral cells, human cancerous tongue cells, have been treated with saffron, and it's found that it induces apoptosis in a dose-dependent manner. Therefore, I hypothesize that if we treat PC12 cells with Hexacyclis aurifolia extract, it will induce apoptosis in a dose-dependent manner. Now, what are PC12 cells and why are we using this line specifically? To answer that, we have to go into a little bit of background story of how this project even started. When I was on my interview with Dr. Sahani, we originally intended to look for a plant that has neuroprotective qualities. The thing about PC12 cells is that they're derived from the medulla of rat adrenal glands. This is interesting because not only are they have a high rate of proliferation, which means that they can divide very quickly. If you treat them with neural growth factors, it can actually differentiate into neuron-like cells, having almost synapse-like structures and whatnot, which would have been ideal for our neuroprotective study. However, upon, like I said, we dug deeper into this plant, it might actually be cytotoxic. So, of course, the first step of this project was to extract oils from the root of this plant. I chose alcohol extraction methods. At first I was debating between alcohol and aqueous, but in the end I went for alcohol because it has a higher yield and relatively faster, and on top of that, my only concern was that maybe the alcohol might affect the cells, but once it dries completely, it shouldn't even be present. Next, and the part that I'm actually looking forward to the most, is the viability test. This is a test that pretty much is going to tell whether the cells are just going to completely die off or not when we give them various concentrations of the extract, and we're going to do it in 24, 48, and 72-hour intervals to see what exactly this extract does to our cells. If they survive, well, then that's just more. That's just pointing towards my hypothesis. The way we're going to do this is an MTT-based cytotoxicity assay, which means that this molecule MTT is going to be administered to all the cells in equal amounts, and viable cells or live cells are able to metabolize this into a different color. We can read that different color and see relatively how many cells are still alive when we administer different concentrations. After that, we're going to go to test to see whether whether it really is saffron which is inducing the apoptosis. And we're going to do this by Western blot. Now, a Western blot is basically just a measure of concentration of different proteins, and this is done by isolating the protein, then treating it with antibodies, which gives a sort of, there are different methods. You can do fluorescence, you can do certain types of other things to it, but this depiction that you can see right here is what generally we should see as increasing concentrations of this extract are administered. The protein that I have chosen is caspase 9, as you can see here. Safrole works through a caspase-dependent pathway, or induces apoptosis through a caspase-dependent pathway. I chose caspase 9 because it's relatively close to the end of the pathway, and literature does say that this protein is, there's increasing amounts of protein with increasing amounts of safrole. So, I guess typically what we like to ask ourselves a lot is, what if I'm wrong? What if my hypothesis doesn't hold up and it's not supported by the test that I'm administering? Well, in a weird sort of way, I'm actually hoping on it. If these tests, if the viability test especially, doesn't kill off my cell cultures, then it gives me an awesome opportunity to study this molecule which I mentioned earlier, beta-asterone. Beta-asterone is linked to neuroprotective properties in the sense that it allows the expression of brain-derived neurotrophic factor, which is also called BDNF for short. Brain-derived neurotrophic factor is involved in neuroplasticity, which pretty much means it's there for the maintenance and growth of synapses of neurons, which again our pixie controls are ideal for. But I think I know what else you're thinking. What about these other two molecules? It's constituted for almost 20 and 8.7% of the rest of our extract. Well, upon researching these, they're actually mutagens, which pretty much they form like an atom to the DNA and make it very difficult for replication to happen and causing mutations. So really what I'm trying to say is anything can happen here. But that is essentially the beauty of biology and this discipline that we all are pursuing in this room. It really is a mystery, and I can't wait to go through this, do these various tests to find out what's going to happen. Beyond that, I just want to say thank you for your time and attention. And as I receive questions, I shall have my lecture.
Sometimes I dig up a bunch of little brown jug and make myself a nice frothy drink and suck down a bunch of saffrons. I'm not going to keel over or just, what my kids are going to be mutants, but
that's for sure.
Well, according to the studies, it's not even completely linked to humans just yet, but what is known is that, well, the methyluginol that I showed in one of the earlier slides, that and safrole are both known to cause tumors in the liver. But that's, again, hasn't exactly been linked to humans just
but
yet.
I'm not going to keel over
no.
20 minutes with
No.
the toxins in me.
No, you have to, first of all, you have to drink a lot of it. And, these compounds are found in small concentrations in all types of spices and herbs, so it's not unique to this plant.
Can you buy commercially prepared safrole and your other compounds of interest so that you might use those as sort of a positive control?
Yes, and that is part of the plan. However, we're waiting to do the MGT assay in order to find out whether it actually does kill cells. Then we'll know what to order.
Okay.
I have a question.
Yes.
So you said that the safrole induces apoptosis in somebody's cells, right? If you find anything, I just be curious, could that possibly be used to treat cancer cells?
Yes, there is actually a lot of research that they try to link it to anti-cancerous properties. However, again, well, actually no. The in vitro study did show that it kills cancerous tongue, leukemia cells, and cancerous throat cells, so it is possible. If that is the case, then safrole would be an awesome candidate for cultivation of this safrole.